cells at low magnification. And so, again, these are going to be kind of like the spindles on the back of a chair in terms of their shape. You can see where the axon would be coming down from here, and then again, the axon branches off into the axon terminals. And so we have our little terminal buttons that are actually interacting with the muscle cell. So when we look at the model, again, we're coming down, we have our axon coming down, and in the case of the model, it's just got a single terminal button. We can see, you can see what looks like kind of like pepper in there. Those are going to be vesicles. What neurotransmitter is going to be in those vesicles? Does anybody know? It's going to be acetylcholine. So acetylcholine is the neurotransmitter that's going to activate our muscle cells to depolarize. And again, if that depolarization exceeds threshold, we get an action potential in our skeletal muscle cell. And if we get the action potential, then we're going to get contraction in response to that stimulus. So when we look inside of our axon terminal, on our model, we can also see, we see a mitochondrion, and then we see small muscle cells. What's the purpose of the mitochondrion there? What does mitochondria in any cell do? This is where we most efficiently produce ATP, where we have the citric acid cycle and the electron transport system happening. And so it takes a lot of energy to do what we do. When you consider maintaining the sodium-potassium ATPase pump to maintain our resting membrane potential, all of our transport gates for calcium to move calcium and keep the calcium gradient as well, takes a lot of energy. So we have to have our mitochondria present to manufacture that ATP that we need to maintain all of the processes. We also notice that our nerve cell has wrapping around it. What do we call that? This is our myelin sheath, and if it's in the peripheral nervous system, the myelin sheath has to be made up of what type of cells? Schwann cells. If it was in the central nervous system, what type of cells would it be made up of? The O ones? Oligodendrocytes, yep. So, when we go from our nerve down to our muscle, you can see that, as Mr. Hershey pointed out when he was looking at the other models, you've got invaginations in the muscle cell, and this increases surface area of the muscle cell. And again, that's important because we're going to need lots of proteins to form our channels, and we're going to need lots of proteins to act as receptors for acetylcholine in the membrane. And the more protein we have in the membrane, the stiffer the membrane gets. So we need lots of surface area to be able to have those proteins. And then we move from the sarcolemma again into the myofibrils. And again, on this model, we see our T-tubule system. Again, we see lots of mitochondria because it's going to take a lot of energy for muscles to relax. And that's kind of the cool thing that happens with skeletal muscle. We need the energy for the relaxation side of things, not the contraction side of things. We use ATP to break apart the contraction and relax our muscles. And that's why when you exercise a whole lot, if you run the muscle cell out of ATP, it cramps. Because you've contracted it, but you can't relax it without ATP. And again, when we look at our myofibril, your last picture in your lab manual, in your lab for today, shows you the different regions along your structure of the sarcomere. And the sarcomere is going to be the functional unit for contraction. And so we talk about a sarcomere being from one Z disc to another. So the gray bands on this model represent the Z discs. Then we have actin and myosin proteins. So our little blue stripes that are attached to the Z disc represent the actin, and then the thick filaments in the center represent the myosin. And again, when we contract the muscle, we're going to shorten the Z-disc. When we relax the muscle, we can expand the Z-disc. And we have the other models that show these as well. It shows you how this is going to work when contraction and relaxation happens. And this is why we talk about the sliding filament model as our description of muscle contraction, because of the way they slide over top of one another. Did Mr. Hershey show you the other slides? If we increase our magnification, we can also see some of the striations in our skeletal muscle cells. And so when we look at the histology of our muscle cells, we're kind of looking for the different visual characteristics to kind of clue us in to what type of muscle cell we're dealing with. And so we talk about things like striated versus non-striated. We look at the shape of the cell, whether we're dealing with a spindle shape or branching cells. And then the presence of intercalated discs and other structures help us identify the different cell types. So when we look at our skeletal muscle, you'll also see it identified on the slides as striated muscle. So where the slide that is striated equals skeletal muscle, although it's not the only striated muscle. So as I move through the depth of field, you can again see the light and dark banding coming in and out of focus. And again, that's because of these overlapping proteins in the skeletal muscle cells. What other characteristic do we expect to see in skeletal muscle? It's multinucleated. So if we look along the length of the fiber, we're going to see lots and lots of nuclei within the cell or along the edge of the cell. We can have as many as 7 to 8 nuclei per cell. We're also going to see the elongated organelles, the mitochondria, because we've got lots and lots of mitochondria to help us provide the ATP that we need. So the nuclei tend to be very short and round. You can frequently see a nucleolus in them. The mitochondria are going to be much longer, and you'll see sometimes the inner membranes weaving back and forth within them. And so our skeletal muscle is going to be striated, it's going to be multinucleated, and the cells are spindle-shaped. When we look at our smooth muscle, the cells taper at the end. They're not striated, and they tend to form large sheets within the walls of different organs and blood vessels. They're typically going to have a single nucleus. And in many cases, because of the way the walls of the organs work, we may have interlapping layers. And so we can see some of our cells more longitudinally here. Other ones we see that are more of a cross-sectional view of them, the way the different cells interweave. When I increase the magnification to high drive, so we're looking at 400 times magnification now, We can again see the round nuclei and the elongated mitochondrion within the cells. But again, we see absolutely no striations. We see a little bit of fibers in there, but absolutely no striations. The proteins in smooth muscle are organized differently than those of skeletal muscle, so they lack the striations. The last type of muscle that we look at is the cardiac muscle. And the cardiac muscle takes us back to a striated muscle, but they're not nearly as well defined as the striations in our skeletal muscle. When we look at it on its lowest magnification, we're also going to see a lot more blood vessels interspersed throughout. We can see a blood vessel here, blood vessel there, smaller blood vessels there. The heart gets preferential treatment when it comes to oxygenation. And so, even though the heart muscle is forming a sheet, the cardiac muscle forms the wall of the heart, and the cells intertwine to form a sheet. There's lots and lots of blood vessels embedded in them. Couple more here and here. And so those are important to provide the oxygenation that those cells need, again, to make all the ATP, because we certainly don't want cardiac muscle to cramp. That would be a really bad thing. So when we get to our high drive power, again, if I change the depth of field focus, you can see some of those striations coming in and out of focus, but they're not nearly as clearly defined as the skeletal muscle. You'll also notice that some of our cells branch. So we have a nice branching cell right here. It's kind of like a Y shape there. That only occurs in cardiac muscle. Neither skeletal muscle nor smooth muscle cells branch. The last thing that we'll see are these little lines here. It looks like somebody drew on the slide with a pencil. Those are what we call intercalated discs. And the intercalated discs are where the cell membranes of two cells actually meet. So they invaginate. What do you think this does? It's going to strengthen the connection because we have an increased surface area connecting the two cells. So it's going to help strengthen the connection. We're also going to see within this lots of gap junctions. Anybody remember what gap junctions are? These are proteins, channels, that span both cell membranes. So whatever happens in one cell directly affects whatever happens in its neighbor. And this is going to be very important for increasing communication. So the cells can essentially, quote unquote, talk to each other better. And this is going to allow the heart to work as a functional unit. It's all going to contract simultaneously in response to a single stimulus to pump our blood. And we'll talk about that a whole lot more when you get into ANP2. Right now, you just want to be able to recognize this as cardiac muscle and be able to identify those intercalated discs on your slide. And they're highlighted in the picture in your lab manual as well. Any questions about those tissues?